Okay, we're still in gravimetric stoichiometry. I would title this one, though, Lesson 2, because we're actually going to take a look at some applications and things that we would do lab-wise and so on and so forth with this stoichiometric method. So this is our first chance to do that one. Some of the stuff that you guys already know, you've had to see it and do it in previous labs and previous things back in grade 10 and in the beginning of Chem 20. And so what we're going to do here is look at testing the stoichiometric method. And we're taking a lot of time here, so this is something that should work. But one of the best things that we can do to test a method is to test a prediction of said method against a well-known standard or target value, to see how well or how good a prediction is. We can test it for its validity with a percentage difference calculation. This is sometimes called percent error. I don't like using that term here at the high school level because we still think error is bad. Now, mistakes happen. The best part of science is trying to correct them or figure out why they happened. So percent difference just means that you're different from a target. Percent error means that there's a certain error limit for that target. So none of these things are bad. They happen all the time. We will present labs to you that have high percentage differences, and we'll have ones that are good. It'll be our job to try and figure out how to fix them if they do go poorly. Excuse me. So, here we have the percent difference calculation. This is in your earlier on in your lab book. It's in the appendix of your textbook. Here we have it again. Percentage difference is just the experimental value that you have. This is the one that you gathered in the experiment itself. This is like your lab obtained number from the evidence that you processed. The prediction is usually the one that the law or the idea is saying that you should get. In this case, these are your stoichiometric predictions. Remember, stoichiometry is just numbers on paper with a perfectly balanced equation. That is a predicted model for a perfect situation. When we run the experiment, there's all sorts of other ways that we can get into, including error or difference, just because of the measurement devices that we might use, or the conditions of the lab compared to stoich, and I could go on and on and on. So, know how to do this calculation. We will use this as a test whenever a target is involved. Percent difference, generally 5 to 10%, all right, depending upon the lab. 5% when we get into Chapter 8 for our titrations, but 10% for most of our other labs. Okay, so it is variable based upon the lab. For our at-home learning, I think what I'll do is I'm just going to say, hey, 10%, and we go from there. Now, if you do get a percentage difference greater than 10%, then you will have to do some talking about it in your evaluation. Now, with dry labs from at home, this, of course, will be limited and it will be kind of tough to do sometimes. But things that I want to do to help you out for things that you cannot include is, number one, your reagents and your skills are both good. Okay? We're not giving you bad stuff that's going to fail the lab. And two, you have a procedure that you can follow, which means you can do this lab as well. So, in other words, human or experimenter error is not allowed as a source of error. If you put this down and tell me that it was how you performed the experiment, not something specific, or just, hey, human error, then that's a major loss on your future labs as we go forward. All right, that'll be a major penalty and you will lose major marks. Okay? You are not allowed to say that you are the source of the error because that's essentially telling me you didn't care about doing the lab or care about analyzing the evidence. All right, I hope that's clear. I'm not trying to be a jerk about this, but I am trying to tell you that in chemistry and in science, you are not allowed to be part of the problem. So there are certain things that we can't avoid. Usually these things are very small, okay, but making sure that we don't round early. All right, if we do early rounding, we can carry down the error of the rounding of the last digit. Non-ACTP conditions are definitely an error, but again, a very small one because they're so close to room temperature conditions. A lab we would be doing here to test this is taking a look at how we might filter or dry or rinse something, Are we accidentally washing away reagent, or are we trying to do our best to keep it? All right. Other things that we might run into here are qualitative judgments. If you think back to Chapter 6 and acid-base indicators, you might have to make a determination on the color of a solution. And your lab partner might say orange, I might say red, and we have to come up with a decision on which one is the more appropriate qualitative assessment. All right. And in the lab situation, okay, we do use different relative purities of chemicals, but they're all generally high. Okay, if we go below this, we tell you. All right, but these are all small little errors. So what we will do here is we will use a 10% error limit. And when we're talking about gravimetric stoichiometry, one of the biggest things that we're talking about in total is this idea of a limiting reagent that we've been introducing slowly through some of the previous examples. In all of the problems so far, there's been one thing that ran out first and something that was in surplus or excess. So all of your stoichiometry is quantitative based upon this guy running out. To make sure that it all runs out, what we want to do is make sure that we have a correct amount of excess reagent. Now, don't forget, in industry and in labs, and heck, even in schools, the reagents cost money, so you don't want to be dumping in way more than you should be using. It just takes a small excess to make sure that the limiting reagent is used quickly and efficiently. Our rule here at St. Francis is that you will use 10% extra for your excess reagent, and I'll be showing you calculations as to what this looks like. This is more or less the idea of, hey, I want this reaction to run in 5 minutes rather than 5 hours, so I want to make sure I have enough excess to use all of this to get through the reaction quickly. How much should I use? 10% over the minimum amount that Stoich will predict. The last thing that we'll do is we can test the efficiency of a design, and we can take a look at the percentage yield. For example, stoichiometry says I'm supposed to get 2 grams of a precipitate, but I only get 1.5. Then I have a 75% efficiency, and I might want to look for ways to improve my procedure and my experimental design to do this. So there you go. There's 2 formulas that normally you would memorize, percent yield and percent difference. One tests for validity, the other one tests for efficiency. That's generally how they're used. And you have them here at your reference, so it should make it easier when those questions appear on the Chapter 7 test at the end. But no, you'll have to have those memorized for Chem 30. Okay, that's kind of the introductory stuff here. The rest is going to be moving into examples. For these examples, what I want you guys to do before we get to the next video is make sure that you have all of the balanced equations for any new problem. All right, I'm going to try and speed through these a little bit more so that I'm not doing 7 videos to get through all the examples, and we can get this done in 3. So you know the grade 10 work of coming up with the balanced equations. You can organize the information underneath, and then we can take a look at the example as a solution. All right, so get those balanced equations done for the ones that we need to, and yeah, we'll go from there. So I will see you in the next video with the balanced equation for 1 and 2 ready to go.